and welcome to the Bioinformatics video exploring two resources from the National Center for Biotechnology, or the NCBI website. These two resources are BLAST and PubMed. The NCBI website houses many databases that are relevant to the field of bioinformatics. My name is Jenny Allen, and I am the multi-campus bioinformatics education specialist at Oklahoma University Health Sciences Center. In this video, we will look at two of the different flavors of BLAST, an acronym that stands for Basic Local Alignment Search Tool. We will compare both an amino acid and a nucleotide sequence to a reference database in order to identify what gene and from what species this sequence most likely will be found. We will also explore PubMed, a database housing peer-reviewed articles. If you have previously watched my barcoding video from the lecture I give at Tulsa Community College, the next several slides are repeats from that video. So if you don't zone out, I do hope you enjoy the review. NCBI, which is under the umbrella of the National Institutes of Health, is a centralized bioinformatics hub. The databases housed here are open source or free to use. Scientists from all over the world deposit and share genetic information from organisms they are studying in an effort to enhance other scientific discoveries. An example might be a scientist studying herbal plants deposits his or her DNA sequences into the GenBank database. Another scientist from another part of the world who is working on a drug for cancer can use that information in their studies. In 2015, there were estimated to be DNA sequences in the form of nucleotides or proteins from over 300,000 species deposited in this database. After a quick review of some basic genomic principles, we will use BLAST to access the database, navigate, and recover valuable information on our query sequences. Time to review. Let's begin with the central dogma of molecular biology, which is simply an explanation of the flow of genetic information through a biological system. We know that DNA is transcribed to messenger RNA, then messenger RNA is translated into proteins. DNA is a double helix structure composed of basic building blocks called nucleotides. Nucleotides have three components: a 5-carbon sugar, a phosphate functional group, and a nitrogenous base. The bases are thymine, which is represented by T, cytosine C, adenine A, and guanine G. These building blocks are connected with a covalent bond between a hydroxyl group on the number 3 carbon of the sugar and the phosphate group on the next nucleotide. Because of this order of assembly, there is direction associated with each chain. We have two chains that run in opposite direction, what we call anti-parallel. This chain is the forward strand, and this chain, its complement, is the reverse strand, and they are joined together by hydrogen bonds. Before we begin our first of 3 exercises, I would like to suggest that after watching each one, pause the video and repeat the exercise. I learn much better by doing than just listening. BLAST stands for Basic Local Alignment Search Tool. I mentioned this previously. It is an algorithm that compares biological sequences, either in the form of nucleotides or amino acids, to sequences in a database. This is useful, for example, if you want to see if a gene that has been well studied by scientists is present in an organism you are studying. Maybe you want to know if a gene from one well-studied bacteria that has been associated with the survival mechanisms in extreme temperatures are present in a bacteria you are studying. You would use the tool BLAST. BLAST comes in several different flavors depending on different factors, such as, is your query DNA or RNA, and is your database DNA or RNA. We will begin by using BLAST N. N standing for nucleotides, because we want to find out the best match from a nucleotide database to our nucleotide query. I am going to demonstrate. So first, we are going to open a browser. First, we will open a browser, then a search engine. I used Google, and we're going to enter NCBI. Then we go to the NCBI homepage. We are then going to click on BLAST, then BLAST in. In the query box, we're going to enter a nucleotide sequence. The first time we are going to use DNA, A, T, C's, and G's. I made up a random sequence of DNA, and then I will input it into the query box. The database we are using is the NR non-redundant database. It houses about 65 million sequences from all organisms. I'm going to click on the box that says Show Results in a new window, and then I will click Blast. This page shows the results of our blast. Notice there are a series of colored bars. The colors are associated with the score the algorithm has determined. The score allows us to quantitatively determine if the match is significant. Simply put, the score is determined by assigning a specific score, either positive or negative, to positions in the alignment for matches, accepted mismatches, then mismatches and gaps. Generally speaking, the higher score, the more significant and the more confident you can be that it is a match. So let's scroll down to the alignment with the highest score, the one associated with this blue bar. And this is it. First, we notice that the alignment has 100% identity. Right there. We also are given the bit score. Right here. 40.1 bits. So we have what's called a raw score, and then we have what's called a bit score. Bit scores normalize the raw score, allowing us to compare different alignments from different database searches. We also have an expect value or an e-value, which is the number of different alignments with score equal to or greater than the score that we expected to occur by chance. Simply put, the higher the score, the lower the e-value. Look at our top hit. Here is our query sequence. It was 25 base pairs. And here is the organism that is its match. Here is the name of the gene associated with this hit. Right here, glucose dehydrogenase. And here is the name of the organism that is associated with this hit. I'm going to copy it. I have no idea what this organism is, but someone somewhere in this world is studying it. Now, how do I usually answer most of my questions? I Google it. It looks like our organism is a parasitic wood wasp. And here is the image of our organism. If I was to type in another unique sequence in the query box of my BLAST-N, I would get totally different results. I mentioned earlier that BLAST comes in different flavors. Say you have an amino acid sequence as a query. Recall, RNA is translated to proteins. Proteins are formed from either a single polypeptide or multiple ones. Polypeptides are encoded by codons, or 3 sequential nucleotides, which specify an amino acid. From the bottom amino acid table, you can see the combination of codons for each of the 20 amino acids. This table has the name of each amino acid sequence, its characteristics, but most importantly, the single letter representing that particular amino acid. This sequence of letters is what is inputted into the query box. We will go back to the NCBI homepage once again. We will click on BLAST, and this time, instead of choosing nucleotide BLAST, we are going to choose protein BLAST. Notice in the tab highlighted BLAST P. P for protein is what is highlighted. And now we insert the amino acid sequence into our query box. We have the NR non-redundant database. I'm going to click this box to have it appear in a new window and click the BLAST. Here are results for our BLAST P that we just performed. If we scroll down to our top hits, we will notice that one of our top hits is amylase alpha-2A pancreatic Homo sapiens. It has a 100% identity and a 100% positive, 0 gaps, a score of 98.6, and expect value of 3 e to the minus 24. Amylase is an enzyme that breaks down polysaccharides or starches, and it is mostly found in our saliva and pancreatic juices. Animals need these enzymes to convert polysaccharides to glucose in order to function. Also in the square brackets is the genus species name for man, Homo sapiens. I want to take a look at how the number of matches of amino acids in some of our results compare to our query sequence, which is amylase for man, and how this relates to divergence among other species. Divergence is simply an indicator of how closely or distantly related different species are. I have already gone down the hits list and made a bar graph of the number of matches for different species in our hit list. I inputted a total of 43 amino acids as my query, and from the results had a hit of 43 out of 43 identical, which was our species for man, or our hit for man. Next, I found a hit that had 42 out of 43 matches. Only one amino acid was different. And this species, the second bar, was for the chimpanzee family. Genetically speaking, man and chimps are closely related. Next, let's look at some results for hits with 39 out of 43, 36 out of 43, and 35 out of 43, or another way to look at it, that third bar had 4 mismatches, the fourth bar had 7 mismatches, and the fifth bar had 8. If we take a look at images of these species, we can see that they are genetically more closely related to each other, but more distantly related to our query species. Finally, let's look at the last 2 bars. The next to last one had 32 matches, and the last one had 31 matches out of 43, or 11 and 12 different amino acids than our query sequence. Looking at these images, we see that these species are birds. These 2 hits are closely related to each other, but more distantly related to our query sequence in comparison to the group with the mouse and squirrel species. Interesting how much information about the relatedness of different species can be gained from analyzing the genetic code of species. We have looked at 2 tools on NCBI. Now let us look at the final tool from NCBI that we are focusing on in this lecture. We are going to examine PubMed. For those students early in your educational pursuits, this tool will become a valuable tool for research purposes. Eventually, you will be asked to either write a paper or report on a particular subject. You want a reliable source, and I hate to be the bearer of bad news, but everything posted on Wikipedia is not always accurate. Peer-reviewed articles are a very reliable source for information. For example, a researcher publishes an article with findings and results from experiments supporting his or her hypothesis. Other research in that field must review that article, whether through studying and questioning it, even trying to reproduce the results, and only through approval by this research panel does this article get published. PubMed is a database that houses peer-reviewed articles. So let's go back to NCBI's webpage by clicking on the NCBI icon. From the menu under Popular Resources, let's click on PubMed. So say an instructor asks you to write a report on a bioinformatics application called barcoding. Barcoding is mentioned on the TCC video, so let's key in barcoding. Search query box. You can either click search or just enter, and you will get the results. We have 3,709 articles. This is way too many for me to I don't have time to go through all of these. So I want to filter down my search. I just want to look for articles that have barcoding in the title. So I backspace up next to barcoding, I hit the left square bracket, I key in title, I enter the right square bracket, and this time you can hit search again, or you can do enter. And as you'll see, we have reduced our results down to 1,240. That is still a lot of articles to go through. In the TCC video, I talked about 2 high school girls that did a barcoding exercise on sushi restaurants. They were wanting to know if the fish that they were purchasing at sushi restaurants was what they were actually getting. So let's filter it down. We want to write a report on using barcoding in the fish industry or sushi industry. So in all caps, enter and then fish. I'm going to hit enter again. Now we have reduced our results to 142. This is a little more doable in researching to write an article. We have reduced our number of hits to 142. We do this by filtering. We're telling the computer that we want to find just the articles with barcoding in the title and have something to do with fish. If we scroll down, we can see that we do have a hit using DNA barcoding to track seafood mislabeling in Los Angeles restaurants. We can click on that, and we get an abstract of what the article is about. Read through this and see if maybe we want to do our report on that. I hope you realize what a great tool this is to have in your back pocket. This is the end of the Tools of NCBI lecture, but before I end this video, I want to highlight a wonderful opportunity available to college junior and senior level students. I am part of the OK NBRE program at OUHSC. NBRE stands for the IDEA Network for Biomedical Research Excellence. The IDEA Network is a program designed by NIH to enhance and encourage research in states with low approval rates of NIH grants. In essence, it is a way of distributing research dollars to reach a broader population, leading to innovations in research. One way of encouraging research is through a paid 8-week summer research intern program. Students at these institutions pictured can apply to do research in different fields at a lab located in the state of Oklahoma. And at the end of their internship, they present a poster of their research. If you are interested, I encourage you to visit oknbre.ouhsc.edu. In the menu on the left-hand side, click Student Programs, and you can read about when and what documents are required in order to apply for this opportunity. Also, please feel free to contact me with questions about NBRE, bioinformatics, or barcoding. I have listed my contact information. I hope you have a better understanding of the field of bioinformatics and a greater appreciation of information that can be attained with these tools. Thank you for your time.